Section 15.1, Features of the Animal Kingdom. By the end of this section, you will be able to list the features that distinguish the animal kingdom from other kingdoms, explain the processes of animal reproduction and embryonic development, describe the hierarchy of basic animal classification, and compare and contrast the embryonic development of proteostomes and deuterostomes. Even though members of the animal kingdom are incredibly diverse, animals share common features that distinguish them from organisms in other kingdoms. All animals are eukaryotic, multicellular organisms, and almost all animals have specialized tissues. Most animals are motile, at least during certain life stages. Animals require a source of food to grow and develop. All animals are heterotrophic, ingesting living or dead organic matter. This form of obtaining energy distinguishes them from autotrophic organisms, such as most plants, which make their own nutrients through photosynthesis, and from fungi that digest their food externally. Animals may be carnivores, herbivores, omnivores, or parasites. Most animals reproduce sexually. The offspring pass through a series of developmental stages that establish a determined body plan, unlike plants, for example, in which the exact shape of the body is indeterminate. The body plan refers to the shape of an animal. Figure 15.2. All animals that derive energy from food are heterotrophs. A shows a black bear, which is an omnivore, eating both plants and animals. B shows heartworm, and it is a parasite that derives energy from its host. It spends its larval stage in mosquitoes and its adult stage infesting the hearts of dogs and other mammals, as shown here. Complex tissue structure. A hallmark trait of animals is specialized structures that are differentiated to perform unique functions. As multicellular organisms, most animals develop specialized tissues that group together into tissues with specialized functions. A tissue is a collection of similar cells that had a common embryonic origin. There are four main types of animal tissues: nervous, muscle, connective, and epithelial. Nervous tissue contains neurons, or nerve cells, which transmit nerve impulses. Muscle tissue contracts to cause all types of body movement, from locomotion of the organism to movements within the body itself. Animals also have specialized connective tissue that provide many functions, including transport and structural support. Examples of connective tissues include blood and bone. Connective tissue is comprised of cells separated by extracellular material made of organic and inorganic materials, such as the protein and mineral deposits of bone. Epithelial tissue covers the internal and external surfaces of organs inside the animal body and the external surface of the body of the organism. Animal reproduction and development. Most animals have diploid body or somatic cells and a small number of haploid reproductive or gamete cells produced through meiosis. Some exceptions exist. For example, in bees, wasps, and ants, the male is haploid because it develops from an unfertilized egg. Most animals undergo sexual reproduction, while many also have mechanisms of asexual reproduction. Sexual reproduction and embryonic development. Almost all animal species are capable of reproducing sexually. For many, this is the only mode of reproduction possible. This distinguishes animals from fungi, protists, and bacteria, where asexual reproduction is common or exclusive. During sexual reproduction, the male and female gametes of a species combine in a process called fertilization. Typically, the small, motile male sperm travels to the much larger, sessile female egg. Sperm form is diverse and includes cells with flagella or amoeboid cells to facilitate motility. Fertilization and fusion of the gamete nuclei produce a zygote. Fertilization may be internal, especially in land animals, or external, as is common in many aquatic species. After fertilization, a developmental sequence ensues as cells divide and differentiate. Many of the events in development are shared in groups of related animal species, and these events are one of the main ways scientists classify high-level groups of animals. During development, animal cells specialize and form tissues, determining their future morphology and physiology. In many animals, such as mammals, the young resemble the old. Other animals, such as some insects and amphibians, undergo complete metamorphosis in which individuals enter one or more larval stages. For these animals, the young and the adult have different diets and sometimes habitats. In other species, a process of incomplete metamorphosis occurs in which the young somewhat resemble the adults and go through a series of stages separated by molts, shedding of the skin, until they reach the final adult form. Asexual reproduction. Asexual reproduction, unlike sexual reproduction, produces offspring genetically identical to each other and to the parent. A number of animal species, especially those without backbones, but even some fish, amphibians, and reptiles, are capable of asexual reproduction. Asexual reproduction, except for occasional identical twinning, is absent in birds and mammals. The most common form of asexual reproduction for stationary aquatic animals include budding and fragmentation, in which part of a parent individual can separate and grow into a new individual. In contrast, a form of asexual reproduction found in certain invertebrates and rare vertebrates is called parthenogenesis, or virgin beginning, in which unfertilized eggs develop into new offspring. Classification features of animals. Animals are classified according to morphological and developmental characteristics, such as a body plan. With the exception of sponges, the animal body plan is symmetrical. This means that their distribution of body parts is balanced along an axis. Additional characteristics that contribute to animal classification include the number of tissue layers formed during development, the presence of or absence of an internal body cavity, and other features of embryological development. Figure 15.3 shows the phylogenetic tree of animals based on morphological, fossil, and genetic evidence. Body symmetry. Animals may be asymmetrical, radial, or bilateral in form. Asymmetrical animals are animals with no pattern or symmetry. An example of an asymmetrical animal is a sponge. An animal with radial symmetry has a longitudinal, up and down orientation. Any plane cut along this up-down axis produces roughly mirror image halves. An example of an organism with radial symmetry is a sea anemone. Figure 15.4. Animals exhibit different types of body symmetry. A shows a sponge with asymmetrical or no planes of symmetry. B shows sea anemones with radial symmetry and multiple planes of symmetry. And C shows a goat with bilateral symmetry and with one plane of symmetry. Bilateral symmetry is illustrated in figure 15.4c using a goat. The goat also has upper and lower sides to it, but they are not symmetrical. A vertical plane cut from front to back separates the animal into roughly mirror image right and left sides. Animals with bilateral symmetry also have a head and tail, anterior versus posterior, and a back and underside, dorsal versus ventral. Layers of tissues. Most animal species undergo a layering of early tissues during embryonic development. These layers are called germ layers. Each layer develops into a specific set of tissues and organs. Animals develop either 2 or 3 embryonic germ layers. The animals that display radial symmetry develop 2 germ layers: an inner layer, the endoderm, and an outer layer, the ectoderm. These animals are called diploblasts. Animals with bilateral symmetry develop 3 germ layers: an inner layer, the endoderm, the outer layer, the ectoderm, and a middle layer, the mesoderm. Animals with 3 germ layers are called triploblasts. presence or absence of a coelom. Triploblasts may develop an internal body cavity derived from mesoderm called a coelom. This epithelial-lined cavity is a space, usually filled with fluid, which lies between the digestive system and the body wall. It houses organs such as the kidney and spleen and contains the circulatory system. Triploblasts that do not develop a coelom are called acoelomates, and their mesoderm region is completely filled with tissue, although they have a gut cavity. Examples of acoelomates include the flatworms. Animals with a true coelom are called eucoelomates, or coelomates. A true coelom arises entirely within the mesoderm germ layer. Animals such as earthworms, snails, insects, starfish, and vertebrates are all eucoelomates. A third group of triploblasts has a body cavity that is derived partly from mesoderm and partly from endoderm tissue. These animals are called pseudocoelomates. Roundworms are examples of pseudocoelomates. New data on the relationships of pseudocoelomates suggests that these phyla are not closely related, and so the evolution of the pseudocoelom must have occurred more than once. True coelomates can be further characterized based on features of their early embryological development. Figure 15.6. Triploblasts may be acoelomates, eucoelomates, or pseudocoelomates. Eucoelomates have a body cavity within the mesoderm called a coelom, which is lined with mesoderm tissue. Pseudocoelomates have a similar body cavity, but it is lined with mesoderm and endoderm tissue. Protostomes and deuterostomes. Bilaterally symmetrical triploblast eucoelomates can be divided into two groups based on differences in their early embryonic development. Protostomes include phyla such as arthropods, mollusks, and annelids. Deuterostomes include the chordates and echinoderms. These two groups are named from which opening of the digestive cavity develops first, mouth or anus. The word protostome comes from Greek words meaning mouth first, and deuterostome originates from words meaning mouth second. In this case, the anus develops first. This difference reflects the fate of a structure called the blastopore, which becomes the mouth in protostomes and the anus in deuterostomes. Other developmental characteristics differ between protostomes and deuterostomes, including the mode of formation of the coelom and the early cell division of the embryo. Figure 15.7. Eucoelomates can be divided into two groups, protostomes and deuterostomes, based on their early embryonic development. Two of these differences include the origin of the mouth opening and the way in which the coelom is formed.